Yesterday, what we did that was non-cyclic photophosphorylation, and in which particular electron is primary acceptor, then it passes through the ETS, the electron transport system, and then picked up by PS1, which further a pair of electron gets excited, picked up by PEA, primary electron acceptor, and then ferredoxin, which is the second complex after PS1. And then it results in the formation of NADPH2 by the reduction process. And for this reduction process, from where the hydrogen comes, it is by photolysis of water. Continuous supply of electron from PS2, it takes place, but how the electrons are replaced, because if continuous supply takes place, then what is going to happen, that all the electrons will get finished up. So that supply is also by photolysis of water. This photolysis of water takes place on the inner membrane of thylakoid. Okay, and because of this, what happens, the oxygen is formed, which is liberated into the air. The protons are there. Because of the accumulation of these protons in the lumen of thylakoid on photophosphorylation. Unlike non-cyclic photophosphorylation, which was it was like that, Z scheme. That is why the non-cyclic was like this, and that is why it was also known as Z scheme. But this was non-cyclic photophosphorylation, also known as Z scheme. And one thing more, there two photosystems were there, photosystem 2 and photosystem 1. Unlike that, this is cyclic. Cyclic means in a circle. Okay, that means the electron, once which gets excited, it comes back to its original position. Now here what happens? Again, we have here the reaction center. Reaction center have chlorophyll A. Then around it we have all the accessory pigments. These are accessory pigments which are around it. Okay. Chlorophyll A, the main reaction center. Then we have these accessory pigments around it. Now, because of the light received here, the sunlight received here, a pair of electron gets excited. And that pair of electron which gets excited, this pair of electron which gets excited is picked up by a primary electron acceptor. I'm not going to write it now because I have written it yesterday also 2 times. So this is primary electron acceptor. It is picked up by this pair of electron. Of course, there is acceptor in the natural system to pick it up. So it is picked up by this primary electron acceptor. And from here, it will start moving, the pair of electron will start moving downhill. Downhill means this way. And on the way, it will be accepted by various complexes. Various complexes will accept them, and that system in which the pair of electron is accepted by various system is known as ETS, electron transport system. Now here in this ETS, which is there in cyclic photophosphorylation, there are 4 complexes. There are 4 complexes. So we are going to make 4 checks, 4 boxes. First box, which has the first complex. Second box, which has the second complex, third box, the fourth box, which has the fourth complex. This is the fourth box. So we have a chain of complexes through which the electron travels. Now what are these complexes? Let's name the complexes. The first is ferredoxin. So this pair of electron, after coming downhill, it is received by ferredoxin. The second complex to which the pair of electron travels is plastoquinone. Plastoquinone. The third where it travels is cytochrome. Cytochrome complex. And the fourth one where it travels is plastocyanin. So it travels here, then it travels here. It travels downhill into this electron transport system. Now, when it travels downhill, you know, this is light reaction which is going on. So in the light reaction, high energy compounds are formed. Which high energy compounds? ATP and NADPH. So here, no NADPH is synthesized, only ATP is synthesized at two steps. One is here, ferredoxin step here, this step. I'll write this electron here and I'll make it here. One is at this step where photophosphorylation takes place. That means to ATP or inorganic phosphate is added and it results in the formation of ATP. And the second step where it takes place is at the cytochrome complex level. Here again, ADP combines with phosphorylation of ADP takes place with the synthesis of ATP. With the synthesis of ATP, this ATP is synthesized. So here, twice the ATP is synthesized, but no synthesis of NADPH takes place. Now, after this, when the electron, the pair of electron moves downhill, where does it go? In place of moving to any outer area, this moves again to the reaction center of PS1. As I told you here, just PS1 is there. PS1 is involved. So only PS1 is there. So it moves to the reaction center of PS1. Unlike the non-cyclic photophosphorylation. What happened in non-cyclic photophosphorylation? The electrons received, they were not the electrons which left the this reaction center, they did not come back. In the non-cyclic photophosphorylation, which we did yesterday, the electrons which came out of the reaction center because of excitement, they did not return back. In place, the electrons to the reaction center was provided by photolysis of water. This happened yesterday, what we were studying in non-cyclic. But here, you think any photolysis of water is required to provide electron? No, it is not required, because the electron is coming back to the reaction center. Once it goes out of the shell, it moves through the ETS, the complexes, and then it returns back. So there is no photolysis of water. There is no need of supplying electron from any outside source, because the same electron is passing through a cyclic manner. It is passing through and coming back to the reaction center. So there is no need of supplying electron from any external source. Okay, so this way what happens, if you see the picture, if you see it is going in a cyclic manner. Now, because it is the electron is moving in a cyclic manner, this photophosphorylation is also known as cyclic photophosphorylation. Here, no NADPH is produced, just ATP is produced at two steps. One is at the ferredoxin step, another is at the cytochrome complex. Now, few important points which I want to tell you about this cyclic photophosphorylation is, I'll write the points here one by one. The first important point is, where does it take place? The cyclic photophosphorylation. The cyclic photophosphorylation, this is why, this takes place in the stroma lamella. Stroma lamella. Whereas when we did non-cyclic, what did we study? That takes place in thylakoid. Thylakoid is a part of grana. This takes place in stroma lamella. Now here, as I told you yesterday, photolysis of water was taking place in PS2. But here you see any photolysis of water? No, because there is a continuous supply of electron. So electron from any outer source is not required. So no photolysis of water. At the same time, yesterday when we were doing non-cyclic, we studied that the protons produced were used for the formation of NADPH2. But in this system, no NADPH2 is formed. So again, there is no need of photolysis. So here, the second thing is, photolysis of water does not take place. The second important point is, photolysis of water does not take place. There is no photolysis of water. This is the second point of the cyclic photophosphorylation. There is no photolysis of water. The third important point, as you know here, only ATPs are formed. So you can write down in the third point, only ATP is formed. No NADPH is formed. No NADPH is formed, only ATP is formed. This is the third important point of the cyclic photophosphorylation. Now, the next important point which is happening here is that there is no electron supply from any outer source. There is no electron supply from any outer source. The same electron moves in a cyclic manner and reaches the reaction center. So no, the fourth important point, no electron supply to the reaction center from any outer source. There is no electron supply to the reaction center from any outer source. The same electron is moving in a cyclic manner and reaching the reaction center. So these are the important points of cyclic phosphorylation, and you have to keep in mind. One or two you can write on your own also, because one or two, another point you can write, see, that is okay, but not that important. But of course, to increase the number of points, you can write that there is no NADPH reductase enzyme in the system. And one very important point which we are missing out here is, just one photosystem is involved. In non-cyclic, the two photosystems are involved, photosystem 2 and photosystem 1. But here, I'll write the fifth point here, only one photosystem, that is PS1, participates. just one photosystem participates, and that is PS1. This is important, another important feature of cyclic photophosphorylation. Now, beta, is it clear to you? Cyclic photophosphorylation is clear to you? Any query, anything you want me to repeat in this cyclic photophosphorylation? Just raise your hands or send me a message so that, and if it is clear, that also just let me know. Okay, Yashika Bhardwaj is answering. It's clear. Anybody else? To any query, any question in cyclic photophosphorylation? Yes, beta. Ananya is yes. Ananya also says it's clear. Yes, yes, bache. Amrita, Ananya Thakur. Okay, Ananya Thakur, if your connectivity is poor, of course, because of rain, the connectivity is poor. Just find a place in your house where the connectivity is better. Sit in that area. Otherwise, also I'm going to upload this video for your help. So no problem. Even if your connectivity is lost, you can go through the video and learn whatever I have taught today. Okay. So children, I think this is clear today. Rohan Wazir, you are not responding today. Is it not clear? Any query, beta? If this is not clear or any query? Yes. Okay, I think he is not attending the class today. I think the connectivity is poor in his area. He's not in the class. No problem. So children, we have finished with cyclic photophosphorylation. Both the topics are very important from the examination point of view also, because both these topics, many questions are asked from these topics, like photophosphorylation, LHC, light harvesting system, antenna complex, reaction center. These terms are very important, ETS and all, very important, and many questions are asked. And in this week's examination also, you will be getting question from this only. The examination is from the historical breakdown. Your this week's test is from the historical background of photosynthesis plus non-cyclic and cyclic photophosphorylation. Now, beta, I move on to the next topic. Let's study some few things which are related to both the systems. So let's start with an interesting topic, which is the difference between PS1 and PS2. We have done PS1, photosystem 1 and photosystem 2 also. Let's study the difference between photosystem 1 and photosystem 2. So that you come to know about these two photosystems in a better way. Yes. Now the difference between, for that I'll draw a line in between, and this is PS1, which is photosystem 1, and this is PS2, which is photosystem 2. So what is the difference between photosystem 1 and photosystem 2? The first difference is based on the reaction center. So reaction center, in both the cases, their absorption capacity was different. Reaction center absorption peak was different in both. Here it, what was it, and here it, what was it? Can anybody answer this question? Reaction center absorption peak was different in both the cases. Can anybody answer what was that reaction peak in case of PS1 and PS2? Or we can also say it in another terms, that reaction center is this, or reaction center is this. In another word, we can say reaction center is this, or reaction center is this. Yes, I want answers from you all. I'm not getting any response. What about the reaction centers of both the photosystems? Yes, beta? What about the reaction centers of both the photosystems? Yes, I want your answers. The PS1 reaction center is, oh no. Chlorophyll A is the pigment which is present in the reaction center. I'm asking absorption peak, Yashika. Chlorophyll, one student is writing chlorophyll A, which is absolutely wrong because chlorophyll A is not, yes, now you are right, Yashika. But the first number you have written is wrong. Reaction center is in PS1 is 700 nm. It is not 780. So we can also write that reaction center in this is P700. And the reaction center absorption peak is 680 nm in PS2. And we can also write it is P680. Is it clear, beta? Is it clear? This reaction center point is clear to all of you? Reaction center absorption peak, yes. Reaction center absorption peak in PS1 is, okay, beta, I have got the answer, is 700 nm in PS1 and 680 in PS2. We can also write it another way, that reaction center is P700 in case of PS1, reaction center is P680 in case of PS2. Another point of difference. You know, when we did cyclic and non-cyclic photophosphorylation, we saw that PS1 is there in both the types of photophosphorylation. But PS2 is there only in non-cyclic photophosphorylation. So we can write the second point, participates in both cyclic and non-cyclic photophosphorylation. This is the second point of difference between the two, because PS1 is seen in both the cyclic and non-cyclic, whereas PS2 it participates only in non-cyclic photophosphorylation. Participates only in non-cyclic photophosphorylation, does not participate in cyclic photophosphorylation. So this is second point of difference. The third point of difference is their location, where these are located. The PS1 system is located, you know, because this was participating, you know, PS1 participates in non-cyclic also, and non-cyclic takes place in thylakoid. So PS1 is located both in thylakoid as well as stroma, whereas PS2 participates only in non-cyclic, and non-cyclic takes place in thylakoid. So PS2 is located only in thylakoid. So the third point, I think you are able to understand, or shall I repeat it? I'll repeat it. See, PS1 and PS2. This is to make you understand. They both together participate. Together they participate in non-cyclic. Together they participate in non-cyclic. And where does this non-cyclic present? It functions at, it takes place at thylakoid. That means PS1 and PS2 will be present at thylakoid. But if we talk about cyclic, PS1 participates alone. When it is alone, it participates only in cyclic photophosphorylation. And I wrote that cyclic photophosphorylation takes place in stroma. So it takes place in stroma lamellae. I wrote, if you remember, that cyclic takes place in stroma lamella. Now you can easily find out that this PS1 is present both in thylakoid as well as stroma, whereas PS2 is present only in thylakoid. So this makes a third point, that PS1 is located both at thylakoid as well as stroma. So the third point of difference between PS2 and PS1 is located both at thylakoid and stroma lamella. And where is this located? Located only at PS2 thylakoid, because PS2 participates only in non-cyclic. So it is located there. The next point is about the photolysis of water. Okay. Now here, and the release of oxygen. You know, as a result of photolysis of water, oxygen is released. So you think in PS1, oxygen, PS1 basically participates in the cyclic. Okay, when it participate in non-cyclic, there it doesn't participate in the photolysis. Photolysis of water takes place in PS2 system, which I told you very clearly. Photolysis of water takes place in PS2 system, not in PS1 system. So you can write down the next: complex for photolysis absent. Complex, there is a complex which helps in photolysis of water. Complex for photolysis of water is absent. And here you can write, because PS2, as I told you, photolysis of water takes place. So it is, the next point you can write, this complex is present. Now, because photolysis of water is not taking place here in PS2, so no oxygen is released. No oxygen is released, or no oxygen is liberated. Here, the next point is oxygen is liberated, because photolysis is taking place. When photolysis takes place, oxygen is liberated, whereas here no oxygen is liberated. The next point of difference between the two is the electron, after exciting, comes back to the same reaction center. So no electron supply from any outer resource. So no electron supply from any outer resource. And here, yes, there is electron supply from outer resource. And what is that outer resource? That outer resource is photolysis of water. Because of photolysis of water, the electrons and protons and oxygen is formed. The electrons which are formed, they reach the reaction center. So there is electron supply from a outer source. So here, these are the differences between PS1 and PS2. I think you are able to understand it. Yes, children, is it clear to you? Photolysis, the difference between PS1 and PS2. Anything you want to ask in this? Okay, clear. Fine. I think these points are very clear to you. I have distinguished them very clearly. So I think you will be able to understand. We come to the next topic. The next topic here is now here, for your convenience, I would like to tell you the difference between cyclic and non-cyclic photophosphorylation. So our next point of difference is between cyclic photophosphorylation and non-cyclic photophosphorylation. So the difference between these two, difference between cyclic and non-cyclic photophosphorylation. Now here, if you saw in cyclic photophosphorylation, both are involved in with the formation of high energy compounds. But when you saw in cyclic, only ATPs are formed. So the first difference you can write down, only ATP is formed. At two places, you know, at two steps, ATPs were formed. Here, both ATP plus NADPH is formed. ATP plus NADPH is formed. ATP at the cytochrome level and NADPH after at the ferredoxin level. This in non-cyclic, both high energy compounds are formed, whereas in cyclic, just one high energy compound, which is ATP, it was formed. Now, the second point of difference is, as you know, in cyclic photophosphorylation, there was no photolysis of water. Because there was no photolysis of water, no oxygen was produced. ATP, oxygen not produced, because there was no photolysis of water. So oxygen was not produced. Whereas in this case, O2 liberated. Because photolysis of water took place, so O2 was liberated. This was the second point of difference. Now in cyclic photophosphorylation, let's talk about the photosystems. Let's talk about the photosystems. In cyclic photophosphorylation, just one photosystem is required. So only PS1 is required. Whereas in case of non-cyclic photophosphorylation, PS1 and PS2 both are required, and as a result you would have seen Z scheme, which is responsible for the formation of Z scheme. So both the photosystems are required in case of non-cyclic photophosphorylation. Now, in case of cyclic photophosphorylation, no external source of electrons is present. No external source, because the same electron circulates and comes back to the reaction center. No external source of electron is present, as the electron circulates and comes back to the reaction center. Same electron circulates and comes back to the reaction center. Whereas external source is present in this case. External source of electron is photolysis of water. This is photolysis of water. Because of photolysis of water, electron is synthesized, and that electron is provided to the reaction center so that there is continuous supply of electrons. So this is the fourth difference between the two. Then another, as you know, the cyclic phosphorylation, in this oxygen is not produced because no photolysis takes place. When oxygen is not produced, then what happens? Because oxygen is not produced, so this can work, this can function in anaerobic condition also, can function in anaerobic condition. I'll write it here, can function in anaerobic conditions, because oxygen is not produced, no photolysis is taking place. Whereas this can function only in aerobic. Oxygen is produced because of photolysis of water, so can function in aerobic condition. can function in aerobic condition. This is the fifth point. And we come to the last point of this is this cyclic photophosphorylation can work in low intensity of light. Can work in low light intensity. If it works in low light intensity, it doesn't mean that non-cyclic will work in high light intensity. The high light intensity denatures the enzyme. So they work in an optimum intensity of light. This non-cyclic, it functions in, works in optimum, which is moderate types, optimum intensity of light. So this is the difference for you between the cyclic and non-cyclic photophosphorylation. And by this, we finish with the topics related to photosystems, photophosphorylation, and till here, you will get in your test, which is going to be conducted on Monday. So, beta, any query in this? Any questions? Whatever we have done till now in today's